For the first time, scientists have reconstructed the mitochondrial DNA from a European woolly rhino, but they didn't get it from where you think they did. Welcome back to Paleopedia, and the woolly rhino is a now extinct species of rhinoceros that lived in northern Eurasia during the Pleistocene epoch, going extinct around 10,000 years ago. It's kind of one of those staples of the Ice Age, alongside the woolly mammoth, megaloceros, and cave lions and things like that. As far as Pleistocene megafauna, the woolly rhino is pretty well understood. We have several specimens, including mummified specimens, and we do actually have genomic data of woolly rhinos already, but this data comes from Siberian woolly rhino populations. Up until now, there has been no European woolly rhino genome sequenced. According to one of the authors of this new study, Peter Sieber, it's a little odd that there haven't been any genomic data from European rhinos, considering the rich history and available data from animals during that time. Instead of trying to find this data through fossils of woolly rhinos, they looked elsewhere. Specifically, they looked at coprolite, or fossilized feces. Now that might sound a little odd, but fossilized poop is actually very useful for paleontologists. It contains a lot of potential DNA and just evidence of what the animal life in that time frame was up to. For this study, the team looked at excrement from the now extinct cave hyena, of which the woolly rhino was one of its prey items. They got lucky. This particular set of coprolite that they looked at, they were able to isolate the European woolly rhino DNA and then put together the mitochondrial genome, something that's never been done before. Mitochondrial genomes, or mitogenomes, are the genetic information inside the mitochondria, the powerhouse of the cell, as we all learned. This genetic information is inherited from the specimen's mother and is a lot more resistant to degradation than nuclear DNA, so this is really useful in tracing ancestral history. Alongside the rhino mitogenomes, the team was able to piece together the cave hyena mitogenomes, allowing us to look at the dynamics in these two populations. According to Sieber, the occurrence of variability in the rhino DNA suggests that across the range, the population dynamics were really complex. But of course, they are going to need a lot more evidence and DNA sequences of this mitogenomes to be able to piece together solid answers to the questions that they have. And while the DNA that they sequence shows signs of considerable degradation, they believe that they found evidence of a populational split around 450,000 years ago in the rhino species. The mitogenomes of the European population are distinct from the Siberian rhino population, indicating that they experienced a split shortly after the rhinos appear in Europe. And while the findings help us get an idea of what life was like in the past, this can also be relevant to today's, particularly when we're discussing climate change. According to Sieber, insight into the population dynamics of these now extinct species that may have gone extinct due to, partially at least, climate change helps us prepare today as the planet changes around us to help protect the now extant species that we have. Hopefully that's true, that we can use this information to try and help our current animal species that we still have today as we drastically change the planet's climate. But there is some cool stuff happening in the paleontology world right now.